So, welcome to the course on neurobiology designed for people who are not really neuroscientists or biologists. And with today's lectures, I want to start with intercellular signaling transduction. So this is basically the first step to understand the communication between cells. The content of today's lecture will be basic structures of a cell and a neuron. Then we will talk about principles of synaptic transmission, structure and function of the synapse, and we will talk about ion channels, at least about a subgroup of ion channels, the ligand-gated ion channels. So in general, we have to deal with cells, living cells. So a neuron and glial cells, which we find in the brain and in the nervous system, function and work in their principle like other cells. So we have structures like the nucleus, which contains the genetic information where mRNA is synthesized. Then we have ribosomes, which are these small little dots here, which are important for protein synthesis, so to translate the information which is stored in the nucleus into proteins which help the cell to work. Then we have the Golgi apparatus and the endoplasmatic reticulum, which both are important for normal cell function, for transport and modification of proteins, and also for buffering systems. In addition, we have mitochondria, which are cell organelles which are important to synthesize or generate the energy for the cells, and this is a special need in neurons who have a very high demand on energy. And the entire thing is swimming basically in cytoplasm, which is a liquid, consists of water, salts, ions, proteins, and it's encapsulated by a cell wall, a bilayer, which basically contains a number of proteins as well. So this is the general basic of more or less each living cell. However, neurons are much more complex. So this is a drawing which is very, very old. However, it already shows us the main constituents of this specialized cell called neuron. So we have the cell body, and from this we have extensions which are called dendrites, which are basically working like the antenna for the cell where the signal comes in. And then we have a smaller structure which is called axon, which is basically the way which is used to send information from the cell onwards. So if we look on a little bit more detail, again we see the cell body, which contains the nucleus, which contains the endoplasmatic reticulum and the Golgi apparatus, which we talked about just before. We find mitochondria. Those are not only found in the cell body, but they are also found in the axons and in the dendrites, because energy is needed all over the cell. Then we find the dendrites about here. So in this case, this cell has a number of dendrites, and these dendrites branch and therefore can form a large network to collect a number of information from different other neurons. For the axon, we have the so-called axon hillock, which is a larger structure which is important to build up the action potential, which you will hear later on in this course, which is basically the sending of information along the axon. And the axon then innervates some other cells through structures which are called synaptic terminals or synapses. So these synapses can form on the cell body, these synapses can form on dendrites, and it's also possible that synapses are formed onto other axons. The position of the synapse is important for the cell because this collects the information and integrates the whole information. The signal speed which runs along the axon depends on what's called myelination, and this is also something which we will deal in more details later in the course. Basically, it's like an insulation which speeds up the signal, which is especially important in several cases. As we know, some of the synapses in the giraffe reach a length of 7 meters. So then, if the giraffe wants to walk properly, speed of signal transformation is important. So beside the neurons, which are basically the signaling and integrating cells, we need some cells which support the neurons. And these cells are called glial cells. So these glial cells are basically cells which sit in between the neurons, which provide a structure, which provide insulation of the whole brain. So we have these ependymal cells, which is a form of glia, which basically separates the brain from other organs, from the environment. Then we have astroglia. This astroglia basically is seen as provider for energy, for nutrients. It basically encapsulates blood vessels and takes nutrients from the blood vessels up and gives it over to the neurons. Then we have oligodendrocytes in the brain. This is equivalent to what's called Schwann cells in the peripheral neurons, and those are the cells which provide the myelination, so they form these sheets around the axons which provide faster signal transduction. And then we have a fourth class of glia. This is the so-called microglia, and the microglia in the end is a type of immune cell. It's basically helping the neuron in case of damage, and it reacts on damage done to the brain. So to get some intracellular signaling and some intercellular signaling, we need a number of channels, and we need a number of signaling devices. The first thing we want to look at is how would a signal run from the cell body to the terminal, so along the axon. So what we see is that it's a row of depolarizations along the membrane. So it's basically an electrical signal. This is done by a number of voltage-gated ion channels. So this is channels which react to a change in the membrane potential and allow the signal to be handed on. And there will be a special lecture on these type of channels. Once the signal reaches the nerve terminal, it finds a structure which is called synapse. In this synapse, we have a typical structure, so it's basically mostly enlarged to get more surface on the interface of two cells. So there's the presynaptic and the postsynaptic part. This is part usually of an axon, and this is mostly part of a dendrite, but it can, as I said, also sit on another axon or on a cell body. In these structures, we again find mitochondria, because we need a lot of energy in the synapse to pump ions, to generate all the things we need. Then we have small vesicles. So this is basically small bubbles which are enclosed by a bilayer membrane, as we find it very frequently in living cells, and they are very specialized things. They carry proteins which allow to pump neurotransmitters from the cytoplasm into the vesicle. They can store neurotransmitters and basically form a storage pool of vesicles which more or less are ready to send on a signal upon request. This sending on of the signal is basically controlled by presynaptic proteins which are regulated by mostly calcium, which then comes in through voltage-gated calcium channels upon depolarization. So when the electrical signal comes along the axon, it will find some channels here which can open up and allow the content of the vesicles to be released. On the other side, we find a structure which is called postsynaptic density, indicated in gray. And this postsynaptic density is more or less a very, very complex structure. It makes and forms a scaffold for receptors to be put there, but also of proteins which interact with these receptors, which modulate the function of these receptors, and which allow proper signaling. The receptors which sit in this structure basically collect the information which comes from the signaling molecules, so the neurotransmitters, and basically take this information to the next cell. So what we have in this step, we have an electrical signal which leads to the release of some chemical substances. These chemical substances, called neurotransmitters, can flood into the cleft between two parts of the synapse, the presynaptic and the postsynaptic membrane, which usually are on two different cells, and then are collected by receptors, and these receptors then give the signal on to the next cell. There are two different types of such transmitters. First of all, we have small molecules. These are very small molecules in terms of amino acids, fragments of amino acids, purines, and they basically have a very fast transmission. They provide a very fast transmission. They can be synthesized in the axon terminal, and they can be refilled into these synaptic vesicles. And the synaptic vesicles which contain these small molecules are called synaptic vesicles, small vesicles, and basically they can recycle on a fast level, and this is necessary for a fast signaling sequence. So if you need to signal from one cell to the other more frequently, then you need these signals. And these signals can be terminated very fast. So it's a very fast and precise type of signaling. So the second class of neurotransmitters are neuropeptides. These neuropeptides are very small proteins, and as proteins, they are synthesized in the cell soma. So the synthesis starts basically in the nucleus with the transcription of an mRNA. This mRNA is then translated into a pre-pro hormone or in a pre-pro neuropeptide during the synthesis in the ribosomes. These newly formed proteins are delivered into the endoplasmatic reticulum. So this is a structure in the cell body which is important to sort such proteins and to process such proteins. From the endoplasmatic reticulum, it's delivered into the Golgi apparatus, which again is important for sorting these precursors of the neuropeptides into the vesicles, into the large dense core vesicles as they are called, together with enzymes which are necessary to process the hormones into functional neuropeptides. The term large dense core vesicle comes from the fact that these vesicles are pretty large in electron microscopy and they have an electron dense core. So basically they look very dark, and this comes from a high calcium content. And this makes it easy to separate them from the synaptic vesicles in EM. During the transport of these large dense core vesicles along the axon, the prohormone convertases and other enzymes which are important for the maturation of these neuropeptides can work and can produce different types of peptides from a single precursor. So these different types of peptides have different lengths, they can have different modifications like amidations. Depending on the state of processing, these neuropeptides of different lengths can have also different pharmacological properties. So the processing is important for the pharmacology of these peptides, so it's important for the reactions they can produce. And this processing differs in different parts of the brain, in different tissues of the body. The synthesis of these neurotransmitters, these neuropeptides, takes quite long. It basically has to start in the nucleus, it needs to be synthesized, it needs to be transported. So they cannot be refilled in the synapse. So if we have a high demand of such neuropeptides, then there will be a lack of these neuropeptides subsequently. But these neuropeptides are not released as readily as the normal neurotransmitters. So they need a much stronger stimulus, a high-frequency stimulation, to be released. This comes from the fact that these vesicles do not sit in the front row of the synapse, but they sit a little bit back, and it takes more calcium, higher levels of calcium, to really fuse them to the membrane. The function of these neuropeptides is mostly not for fast synaptic transmission, as you can imagine. This is more modulatory function. And how this modulatory function can be transmitted, you will see in the second lecture of this series. But the important thing is here for you to remember, we have two different types of neurotransmitters. One are the fast transmissions, amino acids and similar things, so classical neurotransmitters, which can be refilled in the synapse into the vesicles through specialized enzymes which are called pumps or transporters. And we have the other, which is neuropeptides, which basically are more seen as neuromodulators. So if we now look into the synapse a little bit closer, we will see that these vesicles, and in this case I focus more on the synaptic vesicles for fast neurotransmission, we will have some of these vesicles which sit quite far back in the synapse. This is called the reserve pool. So basically these are vesicles which are already filled with neurotransmitter, which contain everything which would be needed for transmission, but they are not on use at the moment. Then we have what's called the active zone of the synapse. This is down here. So this is basically directly at the synaptic cleft. And in this active zone, we have vesicles which are basically prepared to be released immediately. So they are pre-docked to the membrane. They are really there for a fast transmission. And this fast transmission can work in really milliseconds. This is called the readily releasable pool. So they are basically really in the starting grid, and if the signal comes, they are ready to go. Beside this, we have a third group of vesicles. These are vesicles which are basically undergoing recycling. This recycling is necessary to provide and fill this reserve pool, and it works in a way that synaptic vesicles which fuse to the membrane and release their content basically are taken back as empty vesicles from the synaptic membrane, because otherwise the membrane would grow and grow and grow. And as they contain transporters to transport all the neurotransmitters needed into the vesicle, they can take up the neurotransmitter from the synaptic terminal, from the cytoplasm. So the neurotransmitters are usually available here. Sometimes they are basically available because they are normal amino acids like glutamate. Others might be synthesized by proteins which are soluble in the cytoplasm and which basically are there in sufficient amount. The cycle of such a synaptic vesicle you see here. So what we have is from time to time we get new synaptic vesicles built in the cytoplasm to refresh and refill the pool of vesicles of stuff which is needed freshly. So we have a trafficking of these synapses, vesicles to the synapse, and on this way, and especially then in the synaptic terminal, they are loaded with transmitter. So this is basically the same thing which happens in the recycling. Then what we get is the docking. This is basically what happens here. So the vesicle is attracted to the synaptic membrane. Then what's called priming is that this vesicle is basically attached closer to the membrane, like here, and all the complex which is needed for synaptic release forms, shapes up, and just waits for calcium to come in. Once calcium comes in, pre-primed vesicles can fuse with the membrane, which is shown here. So what you see is this content of the vesicle is released into the synaptic cleft in a way that the vesicle and the membrane which envelopes the synapse, which are chemically very similar, they fuse and basically the content is released. After this empty piece of membrane can undergo endocytosis. This is a special process which needs also special enzymes and proteins. In this case, it's clathrin, this red T's, which allows to take back the vesicle from the membrane. But in this case, as said, the membrane is empty. And this empty bag then can undergo either recycling, as discussed before, or it can go into other pathways. It can be also used for signaling back to the nucleus in some cases, or to fuse with some clean-up devices of the cell, so-called lysosomes. For the release of neurotransmitters, we basically have two types of release. One is the full collapse fusion. Basically, the vesicle fully integrates into the cell membrane, and you cannot see the difference between the membrane and the vesicle anymore, so it's fully integrated. This is something which is also used to by the cell to replace some compartments of the membrane. And the other is what's called kiss and run. Basically, you have a short fusion, but not a full fusion, a release of the content of the vesicle, and after that, the vesicle reforms very fast, and it's basically the old vesicle which reforms. Okay, if we now look on such a synapse in terms of function, we just discussed how the neurotransmitter can be released. But what happens afterwards? So the release of the neurotransmitter is the first step in signaling. The second step in signaling is basically receptors on the postsynaptic side which collect this information. And as we said, such an information needs to be very precise. So if I want to have a signal which lasts just a few milliseconds, then I have to clean up. So I have to remove this neurotransmitter from the synaptic cleft, otherwise it would permanently be attached to the receptors. So this cleanup is done by so-called transporters in case of gamma-aminobutyric acid, which is the main inhibitory neurotransmitter in the brain. Same happens to glutamate, which is the main excitatory neurotransmitter in the brain. There might be some other things like acetylcholine, which can be cleaved in the synaptic cleft, but all these processes are very, very fast. And the idea of these transporters, which reuptake the neurotransmitter, is recycling again. So as we said, the vesicles are refilled in the cytoplasm, and what these transporters do, they take the transmitter from the synaptic cleft and transport it into the cytoplasm, where it can be refilled into the vesicles. So this spares, of course, a lot of resources, and it facilitates fast signaling. It's not only the presynaptic axon terminal which can do such reuptake. It can also happen here, and this is an astrocyte. So this is one of these 4 classes of glial cells which we have in the brain. These astrocytes can also take up, which is important because these astrocytes sit and protect the borders of the synapses, and so they can help to reuptake and to clean. Now, in the astrocyte, it would be a shame to leave the neurotransmitter there without recycling it. On the other hand, you cannot just release the neurotransmitter as it is into the surrounding of the synapse to be taken up by the axon itself, because that would then act as a neurotransmitter. So what you have to do is to transform it. And this is basically what happens here. So gamma-aminobutyric acid is transformed into glutamate, which is the original amino acid where GABA is formed from. But as we said before, glutamate is a neurotransmitter by itself. So now the cell cannot release the glutamate and deliver it. What it has to be done is to transform glutamate into glutamine, which is a simple amino acid step. And glutamine itself has no transmitter function. So glutamine can now be released by the astrocyte in close vicinity to the axon terminal, and can be transported into the synaptic terminal, so in the presynaptic terminal of the axon. And there, glutamine can be basically with the same steps brought back to glutamate or to gamma-aminobutyric acid. And then we have restored the pool of neurotransmitters. So this is basically what happens presynaptically and what's going on. So what happens postsynaptically? Postsynaptically, I said there's a number of receptors. There are different classes of receptors. In this lecture, we will focus on one group of neurotransmitter receptors, and we have a number of information on this, and the information we got about these so-called ligand-gated ion channels comes mostly from this friendly guy. This is a torpedo ray, and this torpedo ray has a very, very special, unique feature. They have so-called electrical organs. These electric organs we find in other fish as well, like some types of eels, and they consist of very, very dense packed nicotinic acetylcholine receptors, and these are ion channels, ligand-gated ion channels. And what these electric organs do basically is they can depolarize a large field at the same time, which allows the animals to produce currents and which is used for hunting and also for defense. So the electric eel can produce up to 500 volts with these organs. So this is really something strong, and it can be dangerous also for human beings in contact with these animals. Why these electric organs are so interesting for science, and especially for neuroscience, is because these receptors are very, very densely packed. And from this almost crystalline pure receptors, you can learn a lot about the structure. So you can see how they are placed in the organ. And if you go to higher resolution with crystallography, you can also see that it's basically something which has 5 parts, or at least 5 bulges, and it seems to have a hole in the center. So what we can learn from this is a number of structure. Considering that we also have information from biochemistry which gives us the structural information of the sequence of these receptors, we can generate ideas how such receptors might look like. Like this pentameric structure of 5 subunits in a circle fits very nicely to what we see here from crystallography. And there would be a hole in the center here and a hole in the center here, which in the end means that we have 5 subunits, with each of them having 4 transmembrane spanning domains, M1 to M4, and M2 is basically forming this pore. Yeah? If we look in a cross section, we would have such large arms outside the cell, a hole in the middle, and this would be these 4 transmembrane spanning domains which form the part which sits in the membrane. And this part in the membrane is crucial because this part in the membrane can form a pore through which ions can diffuse. So if we look a little bit further, we can see that actually in ultrastructural imaging, we see that this is like a funnel, and this would be one of these pentameric receptors, or also this one. So it looks like a funnel on the outside of the cell, and then it's a denser structure which forms the gate. So the funnel shape, of course, makes sense considering that you have to collect information from outside the cell. So the funnel shape allows the channeling of the transmitter into this part. And if again we look on the transection, we see the funnel up here, and we see the gate forming structure here, and some intracellular structure which is important for anchoring, so to hold the whole structure in place, but also for modifications. So if we look in a little bit more detail, this would be the extracellular part of the receptor. This is the membrane spanning domains forming the pore, and this is the intracellular part. This is the axis of the whole receptor, and this is a single subunit. 5 subunits would give such a shape, and from top view it would be basically the same which we have seen a few slides before. And the binding pocket for the acetylcholine sits somewhere in here, or you can see it also in here. These different subunits, they have different pharmacological profiles. So some of the subunits might react to some modulators, some others might not react. And we have a lot of different subunits available for a number of different receptors. So for GABA A receptors, also for the nicotine acetylcholine receptor. We have a number of such modulators which influence the function. From this structural analysis and crystallography, we can basically also define and find such binding pockets, like if we look in detail on this part, we can define the binding pocket for acetylcholine, and we can define which amino acids in the receptor are responsible for the binding or contribute to the binding. This is, on one hand, important to understand what the neurotransmitter might do to the receptor. It's also important for pharmacology and for development of drugs to see where we could put a drug which could change the receptor in a way we want to change it. And we can basically design such drugs on the computer. So it's not necessary that this holds true in nature, because there is always some things which influence a model and which might not be reproducible, but it gives us a good idea about what might happen with a certain drug on a certain receptor. So what actually happens if acetylcholine binds, we see here. So in this case, you see that the pore is very narrow. Yeah? If the acetylcholine binds into its binding pocket, then there is a conformational shift in the receptor, so it changes from gray to red, and what we see is that this pore widens. So the similar thing you see down here, it's just on a larger scale. We have basically a widening of the pore, and this physical widening of the pore allows now ions to pass through. If we look in this structure model, we see that there is a lot of movement in these receptors. So the binding of the acetylcholine takes place here, but the pore is down here. So what we see is a lot of movement of single parts of the protein. Yeah, we have a movement of this redshift stuff. This arm moves down here, the green move a little bit in here. Through this we get some movement into this part, and the blue indicated M2 transmembrane domains, so the pore forming ones, move to the outside. And this basically gives us the opening of the channel. So these ligand-gated ion channels, which are also called ionotropic receptors, we find them with different specifications for different ligands. So we find them for GABA, gamma-aminobutyric acid, glutamate, acetylcholine, glycine, and others. These receptors are selectively permeable for specific ions. So whether they are permanent for anions or cations is basically done by charged amino acids in the pore, and the physical size of the pore restricts the ions which can go through. For each ligand, we have a variety of such receptors. So as they are usually built up from different subunits, the composition of these subunits can change. Moreover, we have different forms of these subunits. So these subunits are not always the identical lengths of the protein. There are short form, long forms, which can be produced from different cells, which increases the variability. So if we look on the GABA-A receptor, it's a pentameric structure. So it has 5 subunits. And alone in humans, we know 13 different of these subunits. So if you consider how many permutations you can do with 13 subunits to get a pentamer, you have an idea of how much is possible. Now we have to consider that there are also different splice variants, so there are some of these subunits coming in different flavors, a little bit longer, a little bit shorter, which further increases the complexity, which is possible, yeah? It's not that all of these forms are really built in nature and used in nature, but we have to deal with a quite large variability still. Moreover, it's not only the ligand which is important for the signaling, sometimes we need also cofactors. So some of the receptors are not active if they are just alone with the ligand. They need some cofactor to activate. And some of these ion channels also depend on a depolarization to open the gate. So NMDA receptors, which are one of the classes of glutamate receptors, they need membrane depolarization to remove a magnesium ion which blocks the channel. And only on depolarization this magnesium is released and the ion channel is open for flux. These ion-gated channels can be modulated, so it's not that one channel consists of 5 subunits and it under all circumstances has the same type of conductance. So these ion channels can be internalized, so basically taken away from the synaptic membrane, and they can be brought back. So we have a pool of these ion channels which can be modulated. Now, what happens if we open a pore? Basically, if there is no gradient, if there is no difference between inside and outside the cell, nothing happens. So what we need is to see that there is a difference in the distribution of ions along the membrane. So what we have a lot of sodium and chloride on the outside of the membrane, and we have a lot of potassium inside the cell. So if I open a sodium channel, then sodium can flow along its gradient from outside the cell to the inside of the cell. And what we see is that we have along this cell membrane a charge separation. So we have a lot of positive load outside the cell and a lot of negative load inside the cell, which is called membrane potential and which is important for the stability and for signaling. So what actually happens if we open a sodium channel is that sodium fluxes from the outside of the cell to the inside of the cell, which produces a depolarization. And depolarization is basically the signal propagation. Yeah? It sends on the signal, it delivers, it might induce a new accent potential, it might simply lead to signaling. If we open a chloride channel, then negative charge will run from the outside of the cell to the inside. This leads to hyperpolarization because the negative charge which remained outside is now also transported across the membrane into the cellular part, and we get a higher membrane potential. This hyperpolarization basically makes it more difficult to generate an accent potential in this cell. So basically we can silence the cell on this level, or the neuron on this level, by simply making it more resistant to input. If we open a potassium channel, then the positively charged potassium runs from the inside of the cell to the outside because we have much more potassium inside, and this can lead to either a hyperpolarization if the cell is resting, or in case that sodium already fluxed into the cell and we have a depolarization. This is a mechanism which is frequently used by the axons for repolarization, to rebuild the membrane potential. And of course, after this exchange of ions, the ions have to be transported back to re-establish this gradient, because otherwise signaling would be dead. And this transport takes a lot of energy, and it takes special transporters. And that's why neurons need such a lot of energy for transporting and building all these gradients and to stabilize such gradients. So, just to recapitalize what we have seen in this first lecture, it's basically the transsynaptic signaling, which is a transformation of an electric into a chemical signal and back into an electric signaling. And during this process, the signal might be modulated and various signals might be integrated. And that's what you will learn along the subsequent lectures of this course. Thank you for your attention.